All right, so photosynthesis, sunlight, energy. And you'll also see some similar molecules that we saw in cellular respiration and photosynthesis as well, NADs, except this one is NADPH, but similar electron carriers that we saw in cellular respiration reactions.
This may sound silly, but what makes it different?
There's a phosphorus in there. Okay? So, electrons are going to be boosted just like they were in cellular respiration, and sugars are going to be produced. So water is going to be split, hydrogen and oxygen, and then we're going to use hydrogen and oxygen in different ways to create our glucose molecules. Oxygen is a waste, and that's going to escape into the atmosphere. So plants and animals need each other to survive, yes? Because we produce what for the plant? Carbon dioxide, and plant produces oxygen for us. Now, other things produce carbon dioxide, so the plant really doesn't need us. But we do work hand in hand in our environment. So, different parts of the photosynthesis reaction we're going to have to get straight, just like we did for cellular respiration. We're going to do that for photosynthesis as well. So we have multi-steps, similar to cellular respiration. The first part of the reaction is called the light reaction. That's going to be dependent on what? The sun, that solar energy, to convert to chemical energy. And then we have the Calvin cycle. So we're going to use products of that light reaction to make sugars from carbon dioxide during the Calvin cycle. Remember, we had glycolysis and the formation of acetyl coenzyme A, formation of acetyl coenzyme A that then fed into the citric acid cycle. Correct, electron transport chain. So this is a similar type of reaction. So the initial incorporation of carbon, the using of carbon in this reaction from atmosphere, organic compounds, is called carbon fixation. This is sort of the beginning parts of this reaction. If we lower the amount of carbon in the air, that could be a problem. Or if we increase the amount of carbon in the air, that can be a problem. Yes? It disrupts the balance. This statement, so very true, deforestation reduces the ability of the biosphere to absorb carbon by reducing the amount of photosynthetic plant life. So the carbon that we put out into the atmosphere, usually we don't have to worry about it too much, because who's going to pick that up and transform it? The trees, the plants. But when we start taking away more plant life and spewing out more carbon into the atmosphere, that fine balance is disrupted. And that's the problem with us and some of the waste products from modern machine. There's a bunch of different organisms that do this, make carbon dioxide waste. I don't know if this will work. No. I guess that's a no. But as you see in your textbook, again on page 110, you see a very similar type of diagram that we saw when we talked about cellular respiration. So this is basically the road map. We have the light reaction, which feeds into this reaction. Looks very similar to what? The citric acid cycle, same kind of picture in the diagram. The Calvin cycle is very similar to that. The electrons are going to be what? In this case, transferred instead of to the electron transport chain, where are they going to be transferred to? they're going to be transferred to the Calvin cycle. The Calvin cycle, in a series of chemical reactions, will then result in what? In sugar production. So, chloroplasts, little chemical energy factories powered by the sun, going to help convert that sunlight into chemical energy. So, let's start with the light reaction. Energy, sun. It's called radiant energy, and that's the type of energy used. When I look at it, I see wavelengths of light, wavelengths of energy. And we talked a little bit about that when we talked about the spectrum of light, our visual spectrum of light. Full range of radiation in the energy part of sunlight is called its electromagnetic spectrum. I'm going to try and keep this as simple as I possibly can. Okay? But that electromagnetic spectrum is much, much larger than we can see. We can feel some of it. We actually use some of it to do what? Cook. Some animals can see in these different wavelengths, but we can't. What can we see? Yeah, just here, and that's called visible light. So the energy that's emitted from this electromagnetic spectrum of light is much, much larger than we are aware of. Yeah, but those different frequencies are still in our environment. The only ones that we can actually visualize are between 380 and 750 what? Yeah, nanometers. And that has to do with the wavelength, the electromagnetic spectrum wavelength that's being emitted by light. We can only see it in this range. And different colors are visualized within these different wavelengths within the electromagnetic spectrum of energy being emitted by light. Am I making... No, don't make me do that. All right, so 380 to 750. So if there was something emitting a wavelength at about 725 nanometers, what would I, as a human, visualize? The color red. Do you see that? In 600, yellowish, 500, the bluish, 400, purplish. Yes? Okay. Okay. So, when I look at that spectrum of light, many of the different colors or wavelengths of light being emitted, the energy being emitted by light, is being absorbed by these little grana, thylakoids, chloroplasts. Yes? What's being reflected? The green wavelength. And that's why we see green. In 1883, a German biologist by the name of Theodor Engelmann saw that certain bacteria tended to cluster in areas with higher oxygen concentrations. I wonder what those bacteria wanted. Could this information determine which wavelengths of light would work best for photosynthesis? Okay, whatever. Oxygen-seeking bacteria will congregate near regions of algae. And what are algae doing? Yeah. So Engelmann laid a string of freshwater algae cells in a drop of water on a microscope slide, added oxygen-sensitive bacteria to the drop, and then used a prism to create a spectrum of light shining on the slide. So if you look on page 111, you can see what happened in his little bacteria experiment. So where did the bacteria tend to congregate more.
They hung out at the low and the high.
They hung out at the low and the high.
So they hung out more in the low. It looks like there's more on the lower end.
Why didn't they hang out in the middle? Oh, because they were reflecting green anyway.
So, mostly congregated around algae exposed to red, orange, and blue-violet light, and rarely moved to areas of green light. If it was just green light, what would my chloroplasts be doing? Nothing, because can they absorb that? No. And they're not giving off what? Oxygen. If the bacteria want oxygen, they're not going to hang with the green light, because the chloroplasts aren't giving them what they want. Make sense? Do you ever have a grow light? Anybody have a grow light?
Yeah.
What are you laughing at? I have one. I grow basil in the winter. I'm Italian. I need my basil. I have my little arrow garden. Do you have one too? Love that. What color's the light? What color's the light? Huh? What color are grow lights? Purple. Wonder why? Oh.
They're not really.
No, it's just, well, it emits more of that wavelength of light. Is that better for the plant?
If you really want your plant to grow, you
Huh?
put multi-vapor sodium in there.
Well, not necessarily giving off more heat, but that's what the plant uses
I know it is.
with respect to the electromagnetic spectrum of light. Yes? So, chloroplasts absorb light mainly in the blue-violet and red-orange part of the spectrum. So that's better light for me to grow things in because that's what they use more of. Yes? I don't know if this will work. No. All right. I don't know why it's not working, but oh well.
It's being mean to you,
So again, we see that little experiment on page 111 in your textbook. Photosynthesis. Now, different types of chlorophyll inside chloroplasts, and they're going to help with different things. Chlorophyll A is the guy that's going to mainly like that blue and violet spectrum of light. And the red light participates directly in the light reaction. Chlorophyll B is going to mainly hang with or be stimulated by blue and orange light and tends to participate indirectly in the light reaction. What's one of the things I'm making in the light reaction?
Sugar.
Well, eventually I'm going to make glucose through the whole reaction. Remember those little electron carriers?
Oh, the NA.
NADPH. Yes? Some different pigments that you'll find in plants include a group of pigments called carotenoids. Now, these guys, believe it or not, absorb mainly blue-green light, but not enough energy to drive as much photosynthesis. You're actually seeing these now.
We saw them earlier today.
You probably saw them today. Where did you see them?
Lab.
Oh, did you do your lab?
Yeah, with the alcohol wicking.
Here you go. Very good. And what did you use? What kind of plant did you use?
Oh, we used a maple leaf and we used some other.
Green leaf. It was just I'm not sure the species of plant, but it was just a nice green leaf.
Perfect.
Just a little bush mess of
Yep. That's usually what I do when I run that lab. I start pulling out things from the trees outside, or I trim the nice little green plants in the foyer region. So you saw that there's different, just besides the green chloroplast, there's yellow and there's red and there's orange. You saw it today. I know you saw it today, besides in lab.
We see it outside and
Yeah, you see it in the trees.
of course we can't see them now because there was big flies all over.
I know, they don't want you to wander. See it? Some's over there. Some's over there. I can see it. You can't see it.
We can see the yellow.
Why do I see that now? No, because what does this plant do? We live in Maine. What does this do? It's going to hibernate. It takes a lot of energy. It's not going to die. It's going to hibernate. It takes a lot of energy for photosynthesis to take place. So in the winter time, when it gets very cold, what do those plants do? Drop their leaves. As the leaves start to die, what do we start to not see as much of?
Green.
The green, because what happens to the chlorophyll?
It stops producing.
They stop producing it. So then you start to see these guys, the carotenoids, the reds and the oranges and the yellows. So that spectacular colors of fall foliage are due to this yellow-orange light that's being reflected by the carotenoids, those pigments in our wonderful little deciduous trees.
So it's the chemistry which is within the leaf that...
No, it's the chemicals within the leaf.
The leaf that when the sun hits it, it reflects that.
Correct. Correct.
Okay. Starting to understand.
So light behaves as what we call photons. And those photons are what fire off our little receptors in our retina. Again, different energy photons will fire off different receptors. We'll see different colors. Chlorophyll molecule absorbs photons, and those electrons in the pigment are going to gain energy and start to get bounced around. So as the electrons fall back to their ground state, lose energy, energy is released as heat or light. So basically we're exciting molecules when we expose them to light and causing them to bounce around a bit.
Vitamin D?
No, vitamin D is, yeah, it's light energy is going to help your skin produce vitamin D in a chemical reaction. So we see here chlorophyll, ground state. When light hits chlorophyll, we see excited electrons move and then come back to ground state. When they do that, they're going to give off heat and they're going to give off that energy needed to drive the other parts of the reaction.
That's basically their electron.
Correct. Kind of like that when we talked about that. You got it. You get a gold star. So in the thylakoid membrane, chlorophyll molecules are organized with other molecules in what we call photosystems. Similar to the proteins that we saw in the plasma membrane in the electron transport chain. Yes? That kind of carried those electrons and pushed them through to create energy to put together ATP that we could later on break for the energy. So photosystems, hundreds of little pigment molecules that function like light gathering antenna in a photosynthesis reaction. So, a couple of different things in this reaction. We have something called a reaction center. Chlorophyll molecule that sits next to something called a primary electron acceptor. Kind of like that series of pumps in the electron transport chain. Other molecules that are important in this reaction, and we're going to find them in the thylakoid membrane, is energy, ATP, and that guy. That's our electron carrier in photosynthesis. Remember, we had electron carriers in cellular respiration, NADH. In photosynthesis, we have what? NADPH. So there's a what's ATP stand for? Triphosphate. Adenosine triphosphate. That's our triphosphate. And that's our energy molecule, right? We made that during cellular respiration. So these little photosystems are a series of little pigments that help take those moving electrons and run them through them to create these reactions within the chloroplasts. So, two types of photosystems are going to help in the light reaction. We have one that's going to help split water, and we have one that's going to help do what? Use those electron carriers to help with the rest of the reaction. So NADPH has to be produced. Okay, we see something very similar to that electron transport chain, except now we're just talking about electrons bouncing around, being picked up and being brought back to ground. So it's sort of like, do they have oh yeah, they do. On page 114, we see this cute little diagram with guys and electrons and little wheels moving round and round. That's a nice diagram, a visual of what happens during or with the photosystems in photosynthesis. So, we already said that, didn't we? Did we say that? Yeah. So, that electron transport chain, we're going to see it again in this reaction of photosynthesis. It's going to help connect the two different photosystems, and then who's going to get released? Yeah, energy. So we're going to help make energy in these photosystems. At the bottom of the page, we see these photosystems, proteins in the membranes of what? Those little thylakoids, right? That's what we're looking at right here. So all of these little proteins are located in the thylakoid membrane of the chloroplasts. And we're going to see that electrons are going to be transported through a series of proteins very similar to what we saw in cellular respiration. The hydrogen, those electrons being excited by the sun, are then going to be picked up by these electron carriers, the Calvin cycle. So we're going to go through the Calvin cycle and eventually use one of those little enzyme molecules to help me make ATP. Well, correct. Yes. No, those are just little pictures to help us understand chemistry. You don't have little tiny human beings running around in your body. So, water splitting photosystem, NADPH producing photosystem, both are going to work together in the photosynthesis reaction. With the help of photons, we're going to excite who? Electrons, and then those electrons are going to be pushed through these different photosystems to help us do what? Make ATP. And that energy is going to be used to do what? Make sugars, exactly. In that particular cycle. Correct. I see what you're saying. So, Calvin cycle then. Yeah, no, we're starting to think alike. That's a little scary. Functions like a sugar factory within the chloroplast and helps to regenerate the starting material with each turn. So just like we saw in the citric acid cycle, there's a series of chemical reactions that are going to go on to help. And this is one of those things I just like to go through to show you. You don't have to memorize it. But this is the chemistry of the...
Is that the pyruvic acid that it just made?
No, different molecule. PGA is the molecule. So remember, this is photosynthesis.
There's no pyruvic acid?
No. And again, you don't have to remember these, but I just want you to have an appreciation for this. It's the same type of reaction we just learned about. So here's my C6, my base of my glucose molecule. And then who's going to help me get the rest of it? This little enzyme called Rubisco. And that's RUBP. Rubisco. All right, come on. So now we're going into the Calvin cycle. And there we see our little enzyme helping to build our glucose molecule. And in the process, we're going to use energy and move electrons, very similar to what we saw in cellular respiration. Except we're going to do all that to make what? Glucose. All right. Different plants undergo different chemistry at different times. And basically it has to do with where I live. Those guys out there fall into this category, C3 plants. They're going to use carbon dioxide directly from the air, and they are very common and very widely distributed throughout our ecosystem. Now we know the environments that we're going to encounter as a plant are not always going to be the same. Like here we get rain on a regular basis, we get temperature changes, plant has to adapt to that. But how about if I live in very hot, dry weather? Yeah, I'm going to conserve what? Water. Okay, so I learn, if I'm a C4 plant, to conserve water. Remember those stomata in the C3 plants that were underneath the leaf? They tend to be open all the time. But in a C4 plant, what are they going to do when it's hot? They're going to close. And that's going to help to do what? Yeah, conserve water. Because remember, water is an important part of my reaction of photosynthesis. If I lose all my water, then I'm not making sugars. If I don't make sugars, then I don't make energy. If I don't make energy, see ya. The other types of plants in very, very dry climates are ones called CAM plants. So they need to conserve water, and they only open their stomata when? At night. At night when it gets really cool. Because heat is going to cause me to lose more water at a faster rate. So C4 plants, things like sugarcane, are going to undergo their chemistry a little bit different than the C3 plant that we just described. The CAM plant, pineapples, again, are going to undergo their chemistry a little bit different because of their quest to try and conserve water and hold on to water in their climates. Is that?
Pineapple drops off that tree or that plant, right?
Yeah, the pineapple is the fruit to that particular plant. That contains the seeds to make more plants.
But if it grows in an arid environment, dry, how does it create a pineapple, which is...
Because that plant is used. Exactly. It's used to the environment. It knows how to carry on its chemistry most efficiently in that environment. Okay? So you should know the difference. C3, C4, CAM. Different types of plants. There's my photosynthesis reaction, and that's the end of chapter 7. Oh my goodness, we still have more time. What should we do? No, I can't go over the test. Not everyone's taken it. Well, no. After class, we can chat.
How did the class structure do?
I emailed everybody that information. Check your email.
Maybe if the internet worked here, it was.
You have a
Terrible like this.
you have a smartphone. I use it all the time. All right, let's just begin a little bit of chapter 8. I won't cover the whole thing. But I want to talk a little bit. Yep. You didn't print? That's okay. You have it though. And you have now you have the recording to go back to. So if you want to take notes on your PowerPoint, you can do that when you listen to the recording again. So there's my favorite guy again. Cellular reproduction, cells from cells. So the little blurb here, 2002 zookeepers at the Chester Zoo were surprised to discover that their Komodo dragon laid eggs. What was the problem? There was no male, just female. Eggs develop without fertilization in a process called parthenogenesis.
That's the problem with the movie.
Hmm?
That was the problem with the movie.
Jurassic Park.
Oh, I guess I didn't see that.
That produced, the guy got all loopy because when you just produce females, well, I got news for you.
Nature will find a way.
Nature will find a way.
If they would have made all males, it would have been a different story. They didn't, they made all females.
But because of this, yes, DNA analysis confirmed that her offspring had genes only from her. So that can happen. We are sexually reproducing organisms. So we as a species need to have two sexes in order to make a new organism. That's not the case for all cells.
The society doesn't think so.
So Komodo dragon is known to have reproduced asexually. That process is called parthenogenesis. Komodo dragon.
A worm.
Yeah.
Like
Different single-celled organisms. I'm not sure about sponges.
this spud or
They
bud or spud.
polyps, yeah, they.
That's sperm. Yep. So different organisms can do this. Cell reproduction. Why do we do it? Why do you do it? You're doing it right now. Yeah, to replace dead cells. And then we also produce special cells to make new members of our species as well. These are two separate cases, and we'll talk about the difference between one and the other. But reproduction may result in in the birth of new organisms, but most commonly involves the production of new cells. So how many times do you make new organisms in your lifetime? New organisms? Twice. I'm in the 2 category myself, but you're constantly making cells every day. 3.
Well, I actually didn't.
Well, you had something to do with it, that's for sure. So when a cell undergoes reproduction or cell division, we're going to see 2 identical cells. We're basically going to clone a cell. That process is going to give us genetically identical cells, and that you're doing now. What kind of cells are you replacing right now?
Skin cells.
Skin cells. That's a big one, right? Hmm? Those are all dead, actually.
Yeah, dead, yeah, exactly, dead, yeah. Hair,
So those cells that they come from are called parent cells. So before a parent cell splits into 2 separate cells that look exactly like it, what must they do? In order to make something exactly the same,
It needs to be copied.
it needs to be copied.
It needs to be copied in the chromosomes and.
Needs to do a
chromosome needs
drive-by
to be-
chromosome.
Anyway, it needs to be copied.
Well, not really a ribosome, but it needs enzymes to duplicate itself. So we need to make new chromosomes. We need to make a copy. During each division, each daughter cell is going to receive the exact same copy of genetic material that the parent cell had. So in order to reproduce cells efficiently, the first thing I have to be able to do is make a copy of my genetic material. Anybody know what this process is called? Making new cells? Begins mitosis. Mitosis. So cell division plays an important role in the lives of multicellular organisms. Are you a multicellular organism? Yes, you are. So it replaces damaged or lost cells. It's going to help you grow as an organism. And it also allows for reproduction of the species. So this is two different cells. You can see them all on the bottom undergoing cell division. Human kidney cells are constantly replacing themselves. We can see this cell near the end of its division. And we can see there's actually two that are going to form. This is a human embryo. And what do we see there? A whole bunch of different cells being produced from how many cells? One. So you started out as one cell. Two half cells came together to form you. One cell. What came first? What do you mean, as far as a molecule in the environment?
Well, no, cells came from, cell theory is everything.
Cells come from cells.
Cells came from cells. So how, if the cell came from a cell, what came first? The DNA or the mRNA?
What do you think?
But where did the DNA come from?
It's just like the chicken and the egg.
Great. We could sit here and argue this all day, right?
But it is the chicken. They're the only ones that have protein and produce the eggs. So
It goes
therefore,
back
had
to
to big
the
DNA.
DNA.
So goes the
Where'd the chicken come from? Did they have to be a chicken?
evolution.
Yeah.
Evolution.
Yeah. It came to mud.
So the question you're asking is, where did the first cell come from? I don't know. But in order to have a cell, what do you have to have? You have to have DNA and RNA and all of the different proteins. So those had to come first before they got organized into a cell.
Didn't they say that the mitochondria is actually a
Used to be a, yeah, exactly.
that the eukaryote developed and decided they wanted to use?
Or the prokaryote decided it was great to hang out here and didn't want to leave. Who knows? So, different types of cellular reproduction. The mitosis that we're talking about, asexual reproduction. Single cell organism reproduces by simple cell division. No fertilization, egg and sperm, no two different cells to make an organism. Some multicellular organisms, like these listed here, sea stars, for example, can regenerate from different parts of their body. So,
Bring a whole sea star?
well, that's a misnomer as well, because you have to have certain parts intact in order for them to make a whole brand new sea star. Yeah, the central regions, yeah.
So it doesn't do what that thing says.
It does in a way, but it can't. You can't just pick off a little end of a sea star and make a whole brand new sea star. That I don't know. No, it's not just boom.
Kind of like a tail on a lizard. All the time they do that.
Well, and think about cell division. So it depends. So in ideal situations, it can happen faster than non-ideal situations.
Sure that scientists grew human hair on the back of a rat?
I thought so too.
Ah, yes. Yeah.
Yeah, they don't have the ability to make it here yet, but they made an ear grow.
On,
a
yeah,
rat.
they took
Cool.
Say that within like 15 years of science evolves the rate that's going now, that with cell,
Stem cell research.
no, maybe that might have been it, but a kid in one of my classes was telling me about it, that like if science goes the rate that it's going now, then like 15 to 20 years, we'll be able to grow like by planting cells on our necks and gills, breathe underwater, breathe crap like that.
Who knows? I don't know about that.
And
Messing with the God particle.
Google released a company that's designed to find the end of natural cause of death through stem cell research. So basically, if you want to, and you have all the money in the world, you can live forever.
Here we go with the time. How much time you got?
How much time you got?
Yeah. Maybe you seen it. Your regular lifespan ended, but you can buy more time.
Oh, no.
That was a stupid movie.
Oh, no, I haven't seen that.
Yeah,
Well, right now we can't buy more time because when we talk about cell division, we'll see why. But some multicellular organisms can make whole brand new organisms from parts. New plants you've done this. Has anybody done that before? Taken parts of a plant and made a whole brand new one? No? You have. You have. I do that all the time. I have one of those Christmas cactuses that drops. All you have to do is take the stuff that drops and stick it right in the dirt. So that happens all the time. So asexual reproduction has a single parent cell, and its offspring are going to be exactly the same. That process is mitosis, and it's a type of cell division responsible for that asexual reproduction and for the cells that you're reproducing as we speak. So all those cells that you're losing off your skin aren't going to, it doesn't mean that you're not going to have skin in a few more days, because you're constantly making more to come up and replace those dead and dying cells. So it's for growth and maintenance of us multicellular organisms. 46. 23 pairs.
No, she definitely told us that.
I think I told you that during class.
Whatever it was, 36. That's what I thought it was. I got full test from this.
46. I was thinking 27. 23. That's why it's
From each parent. From each.
So sexual reproduction requires fertilization. It requires a combination of genetic material, egg and sperm. Many plants are sexually reproducing organisms. Yeah. When you in the spring, when you have all that pine pollen all over the place, that's sperm from the pine tree, all over your car. Brings it to a new light, doesn't it?
like 3 or 4 times.
So, sexually reproducing organisms use a process called meiosis to make those cells. So there's half the genetic material in a reproductive cells than there is in a regular cell. And the two terms we use are somatic cells, those are our body cells, and then reproductive cells are called gametes. And they have half the genetic material that our somatic cells have. Yes? What's soma mean? Soma. Body. So mitosis for growth and maintenance, meiosis for reproduction. In our eukaryotic cells, of course you are eukaryotes, most genes are located in the genetic material we call chromosomes inside the nucleus. Some genetic material is found in your mitochondria and your chloroplasts if you're a plant, but most of it's found in the nucleus.
Does the genes inside the nucleus have the same
Well, the mitochondria actually do that on their own.
But when the cell divides,
Mitochondria have to do that.
also,
Yes,
so they have to make sure that they're in group.
correct. And that happens during a process called cytokinesis. It's part of the mitosis
It's a certain
steps.
stage.
It happens at the end of anaphase and the beginning of telophase. And that's where you're going to basically reproduce all of your organelles. So each eukaryotic chromosome contains one very long DNA molecule. Thousands and thousands of what? You know, thousands and thousands of... We're going through every organic molecule except for DNA.
Well, one of the things is the black is
What makes up DNA? Nucleic acids.
Adenine, guanine, cytosine.
That's a P. It's a P. I'm sorry.
phosphorus, sugar, nitrogen, sugar, space.
Phosphate, sugar, nitrogenous base. What are those called?
Adenine.
Those are the nitrogenous bases.
Okay.
What's that called?
Key to life.
It's called a nucleotide. Remember that?
Yeah.
And the series and the order of the nitrogenous bases and the nucleotides that make up DNA molecules are what are going to make up our genes. Code. It's code. Just like a computer. I have to read a certain code in order for me to do a certain thing. So the number of chromosomes in eukaryotic cells depends on the species. So we're humans, we're the highest creatures on the planet, right? We're the smartest ones. We must have the most genetic material. Actually, no. This rat down here has 102 chromosomes. How is that possible? Why? What are the chromosomes doing for me? Doesn't have anything to do with size. It has to do with directions. Did you say something? No. Oh, duck-billed platypus. So we have 46 chromosomes. The duck-billed platypus has more than we do. So does the buffalo. So does your dog. It doesn't mean that they are more knowing than we are, although sometimes I wonder. I think my dog has it made as she sleeps on the bed as I get up and go to work, and she's like, yeah, see ya. Feed me and I'll poop in the house if I wanna. I don't know who's got it made. I think the dog does. Chromosomes are made of chromatin. So if I take those nucleotides and I string them all in line, that's chromatin. Remember, DNA is a double helix of all these. And those are things that we're going to use to make what we need to make. Chromatin's what we used when we underwent protein synthesis. Remember what that was? The ribosomes were doing their thing. What was that? There's two parts to that stuff. Making a protein. I had to take down the message, and then I had to transcription and translation. Very good. So the chromatin are the little units that I'm going to unzip for transcription and translation. Those are the things that we're going to use along with some enzymes to help us make our proteins. Is that all ringing a bell? Good. So fibers composed of roughly equal amounts of DNA and protein molecules make up chromatin. And the protein molecules, they're called histone proteins, are used to help keep the DNA in order. Wrap it up nice and tight. Make sure it doesn't fall apart. You can see a nice picture of it on page 124 in your textbook. We don't typically see chromosomes until cell division. So when we look at a cell under the microscope, do we see these big fat X-y looking things? What do we see inside the nucleus? Well, it kind of looks like sand, right? But chromatin. So if you look at that diagram on that same page, you'll see thin strands of chromosome, chromatid, and when I pack it up tight, tight, tight to move it, I see chromosomes. So when you look under the microscope here, you see those big, thick things that look like, I call them spider legs, little hairy spider legs. Well, we're hitting Halloween soon, so it's a good visual, right? That's chromosomes. These guys over here aren't quite packaged that way. These guys, see the difference between that nucleus and this nucleus? This kind of looks like sand, little strands. That's chromatid. Now I'm going to make chromosomes when I start doubling my material and packaging up for the move. When I start undergoing mitosis. Actually, I'm going to double my genetic material just before mitosis. But I'm going to start making the chromosomes during the beginning stage of mitosis. So DNA in a cell is packaged into elaborate little multi-level systems, coils and folds, with the help of little proteins called histone proteins that create little packages of DNA, or chromosomes. So chromatid and chromosomes, what's the difference? Well, chromatid are going to have some histones too associated with them, but chromatid and chromosomes, what's the difference? Well, one's looser, more available, and one's packaged up tight, tight, tight. Nucleosomes consist of DNA wound around histone proteins. I don't know if that'll work. It's not going to work. Come on. Yeah, it doesn't work for some reason. I don't know. I have to figure it out. So this is that picture in your book. This is chromatid. When I start packaging, that was a very difficult word to say just then, really tight, I'm going to see chromosomes. And basically, with the help of histone proteins, I'm going to take two copies of genetic material that I created and stick them together. And I'm going to stick them together with a little sticky protein at this area called a centromere. So when I look at any given chromosome, it's actually two copies of the original chromosome. Why do you think I make it? To split. So when I make my daughter cells, those parent cells make daughter cells, half goes to one, half goes to the other. And that's where I'm going to make my copies of information just before I go into the mitotic stage. All right, if I say anything else, I think you're going to explode. So I won't say anything else. Do me a favor, anybody have a piece of paper? Can you rip it out and everybody who's here today sign in? And then you can be on your way. I will open up quiz number 7. That will be due the typical, you know the drill. And we'll pick up with chapter 8 on Monday of next week.